Bismillahirrahmanirrahim. I would like to share the knowledge about Eimeria. My name is Sarfraz Rahman. I am a PhD scholar in the Department of Parasitology. I would like to share this knowledge with references these books, Veterinary Parasitology, second edition by G.M. Arkuhat, Veterinary Parasitology, fourth edition by M. Taylor, George's Parasitology for Veterinarians, 10th edition by D. White, D. Bowman, A Handbook of Veterinary Parasitology, Domestic Animals of North America by Henry J. Griffiths. And last one is the internet sources. Please subscribe my channel and also push the button of bell icon. This is the brief classification of the Sporozoa, the subphylum Sporozoa. It is also called the Apicomplexa. The Sporozoa or Apicomplexa, it is divided into the three classes. The first one is the Coccidia or elementary Sporozoa. Second one is Pyroplasmidia, and third one is Hemosporidia, and it is also called the blood protosporozoa. So the Coccidia or elementary Sporozoa, it is divided into two main family. That first one is Eimeridae, and second one is the Sarcocystidae. And this Eimeridae family, it has the three main genera. The first one is Eimeria, Isospora, Cryptosporidium, and second one is the Sarcocystidae. This Sarcocystidae family has the following genera: the Toxoplasma, Neospora, Hemondia, and Basnoita. So this class Pyroplasmidia, it has the following genera, that the Theileria, Babesia, Cytoxazone, and the other class, the Hemosporidia or the blood sporozoa, it has the three main that is the genera: the Hemoproteus, Plasmodium, Leucocytozone. So the Sporozoa or Apicomplexa, this what means the Apicomplexa? The Apicomplexa, this group has possession of an apical complex, the structure which apparently assists penetration of the host cell. It is visible only with electron microscope, and having the apical stages in their life cycle. And trophozoites have no cilia and no flagella. Intracellular parasites, the sexual stages, it is also called the gametogony. The stage at which the gametes produce spores is called sporogony, and the asexual stages, it is also called the schizogony. And it is divided into three main classes. The first one is Coccidia or elementary Sporozoa, second one is Pyroplasmidia, and third one is Hemosporidia or blood sporozoa. So this family Eimeridae, these are the intracellular parasites, and schizogony and gametogony take place in same host, and sporogony or maturation take place outside of the host. And these three genera in this family, that the Eimeria, Isospora, Cryptosporidium, they are the Coccidia. This Coccidia, it is the infection of Eimeria or Isospora, or collectively of Eimeria and Isospora. And the important point, this is the most important point, that Coccidia has five main that the group, that is the Eimeria, Isospora, Cryptosporidium, Toxoplasma, Sarcocystis, and these have the different points that Eimeria it has the 4 sporocysts and 2 sporozoites, while Isospora it has the 2 sporocysts and 4 sporozoites, and Cryptosporidium it has the 2 sporocysts and 4 sporozoites. Toxoplasma, it has the 2 sporocysts and 4 sporozoites, and Sarcocystis, it has the 1 sporocyst and 4 sporozoites. So Eimeria, this genera, it causes the coccidiosis, host, poultry, sheep, goat, cattle, horses, rabbits, pigs, and turkeys. The site, the intestinal epithelium, but one species in the kidney and one species in the liver. The species, the poultry, the species Eimeria tenella, Eimeria nicatrix, Eimeria broniti, Eimeria maxima, Eimeria mitis, Eimeria isorvolin, and in sheep, the species are the Eimeria credalis, Eimeria ovinoides, and Eimeria bacoinensis. In cattle, the Eimeria zooirni, Eimeria bovis, Eimeria ella bamensis, and in goat, Eimeria erlo-ingi, Eimeria ninna col laico uvi. In rabbits, the Eimeria flaviscens, Eimeria intestinalis, Eimeria steady, it is present in the liver, while in geese, the species Eimeria incerus and Eimeria monosens, and Eimeria truncata, that is present in the kidney, and in horses, Eimeria leucoalti, and in pigs, Eimeria Diblerki. And in Turkey, the Eimeria meliagrimitis, Eimeria adenoides, and Eimeria tenella, it causes the hemorrhagic fecal coccidiosis in the young poultry. Ovocyst, the size 15 to 50 micrometer, and micropyle, a small pore at the end of the ovocyst. Polar cap, micropyle covered by a cap, and monoxenous life cycle whose development is restricted to only a single host species, and excitation. One trophozoite with 4 nuclei emerges and divides 3 times, and each nucleus divides once to produce 8 trophozoites from each cyst. The terms related to life cycle of Eimeria: the sporoblast, the nucleus of oocyst, divides and makes 4 conical bodies, and each nucleus surrounded a mass. It is called the sporocyst. And the sporocyst, each sporoblast secrete a wall of refractile material is called the sporocyst. And trophozoites, the sporozoites enter into the epithelium is called the trophozoites. Chazons, the trophozoites divides and multiple fission take place is called the chazons. Merozoites, what is the merozoites? It is a structure consisting of many elongated nucleated organisms. Schizogony, asexual reproduction. Gametogony, male and female gametes combine. Sexual reproduction is called the gametogony. Ovocyst, male and female gametes combine and zygote form, and cyst is called the ovocyst. Microgametocytes, that is the female, and in microgametocyte, that is the male gametocyte. So this is the life cycle of Eimeria. IP or PP period is that the 5 days in poultry and 3 to 4 weeks in ruminants. This is the life cycle of Eimeria, that in poultry, in the feces, that is the unsporulated oocyst that changes into the sporulated oocyst, then sporocyst, then sporoblast, and then trophozoites. And in the poultry, these trophozoites enter into the intestine where it changes into schizons, and then merozoites, then again it changes into the schizons, and these schizons changes into the microgametes and macrogametes, and then it changes into the zygote. The infective stage is the trophozoites. Coccidiosis in different animals. The coccidiosis in poultry, turkeys, cattle, sheep, goats, horses, geese, rabbits, and pigs. So avian coccidiosis and intestinal coccidiosis, that is the most important from the other coccidiosis. So the species which are involves, that the Eimeria nicatrix, Eimeria iservolina, Eimeria maxima, Eimeria mitis, Eimeria proecox, Eimeria hagani, Eimeria mewiti, Eimeria bruneti. And host, that is the chicken, and habitat Eimeria nicatrix, interior or mid portion of the gut or small intestine, and prepatent period at 4 to 7 days. Eimeria bruneti, the lower small intestine, rectum, cecum or cloaca. Eimeria iservolina and Eimeria mewiti and Eimeria hagani. Eimeria meatus and Eimeria proecox, upper half of the small intestine. Eimeria tenella, that is present in the ceca, that preparent period is 7 days. Identification: the species of Eimeria and the small intestine differ in the size and general morphology. For practical reasons, they can be you can use it and identify it. Approximate size of the oocyst in the microns or Eimeria nicatrix, that's 20 multiplied 17, Eimeria iservolina 17 multiplied 14, Eimeria maxima 30 multiplied 21, Eimeria metus 16 multiplied 16, Eimeria proecox 21 multiplied 17, Eimeria hagani 19 multiply 18, Eimeria mewiti 16 multiply 13, Eimeria bruneti 24 multiply 20. So the distribution and importance: North America and worldwide. Each, each species varies in its ability to produce a disease life cycle, like the Eimeria tenella, and transmission, ingestion of contaminated food or water. So the clinical signs, that the soft feces containing blood, dullness and listless, and drooping feathers, poor weight gain and food conversion rate, decreased feed intake, increased water consumption, weight loss, catarrhal enteritis in thickened intestinal wall, and intestinal lumen filled with clotted or unclotted blood, petechial hemorrhages are present, intestinal epithelium slough and be replaced by connective tissues, interfere with the intestinal absorption, and some species seem to be provoked. A catarrhal and necrotic enteritis and coagulation, necrosis, circumscribed white spots seen through the mucosa. So the diagnosis: on the basis of post-mortem examination, you can identify fecal examination. Pathogenic effect occurs before oocyst production. Presence of large number of oocysts are not necessary for clinical signs. At necropsy, based on demonstration of coccidial forms from the characteristic lesions in specific locations in the intestine. Coccidial forms are not always easy to demonstrate. Treatment: sulfadimidine, that is the drug of choice, and 100 milligram per 200 liter of the drinking water for 3 days. Sulfaquinoxaline, diiodine or sulfadimidine in poultry, 125 ppm in the feed for 8 weeks. So control, it is very important that the good management and use of anticoccidial drugs. Litter should be kept dry. Feeding and water utensils away from the litter. Good ventilation reduce the humidity. Switch program, that the drugs are switched between the batches of broiler poultry, and shuttle program, that is the drugs are switched within the lifespan of the broiler batch, and replacement of laying birds. Preventive medication: sulfonamides, nitropurazoles, nicarbazine, pyrimidine derivatives, and others. The vaccination has been practiced with the concurrent use of sulfonamides. Okay, thank you so much.